This is a video review of Module 1 Quiz Part B, and I'm going to review the questions most missed by the class. Question 1 presented loose connective tissue, and this is the specimen that was presented. It's a loose connective tissue, and if you increase the magnification, you could see there are collagen fibers, you could see the nuclei of fibroblasts. So what is the dominant cell? When in loose connective tissue, the dominant cell here in this view is a fibroblast. The next question most missed was question 2. It's identify the tissue that borders the lumen, and the correct answer was transitional epithelium. And this is the specimen here. There's a specimen of kidney down here in the pelvis of the kidney, and here is the epithelium, and you can see it's stratified. This is the lumen, so this is the surface, and these are very large dome-shaped cells at the surface. Now, some thought that these were stratified cuboidal, but they're not. They're dome-shaped, and some thought they were stratified squamous non-keratinized. The cells would be flat at the surface if that were the case. Okay, so the next question is question 6, where you are asked to identify the tissue, and the correct answer was stratified squamous non-keratinized epithelium. Now, many in the class thought it was transitional epithelium, 25% to be exact. So this is the specimen that was presented. And you can see the stars are present here and the line, so it's the tissue between here. If you magnify this, you can clearly see that, of course, it's stratified without question, and the surface cells are very flat, so it's stratified squamous, and you do not see any keratin, and all the cells at the surface have a nucleus, so it's stratified squamous non-keratinized epithelium. Question 8. Identify the tissue between the two dashed lines. The correct answer was stratified squamous non-keratinized, but 11% of the class said it was, keratinized. About 11% of the class felt it was non-keratinized. This is the specimen here. This is actually a specimen of skin. You can see the epidermis, the dermis, and the hypodermis. And if we magnify this and go right to the surface, then this is the epidermis, and it's stratified squamous keratinized. You see the reddish stained protein keratin, and it lacks any nuclei. I can enlarge this even more artificially, and you can still, here's the nucleus here, but there are no nuclei present here. So we'll go back to this. And just as a way of review of the skin, this layer immediately under the epithelium, under the stratum basale, where you can see several melanocytes, or they also could be Merkel cells here. Here is the papillary layer of the dermis. And down here where the connective tissue is thicker, this is the reticular layer of the dermis. Okay? Okay, question number 9. Ask you identify structures. 2 identical structures were indicated, and each structure has a narrow slit, and the correct answer was stratified cuboidal epithelium. Now, 14% of the class felt it was stratified squamous non-keratinized epithelium, and then we had a distribution of others incorrect answers. This is the specimen here, and that's actually a specimen of skin, and this is in the hypodermis. It's actually just below the reticular layer of the dermis. You can see that here. And so here's the location, and if you magnify this, you can clearly see that there are 2 layers of nuclei around a narrow slit lumen. So that has to be stratified cuboidal epithelium. Question 10 asks you to recognize certain cells, and you're asked what they produce. So the specimen again, was a specimen of skin. Here is the thin epidermis up here, the dermis, and then the hypodermis, or the panniculus adiposus, where you will find glands and connective tissue. But the location is right here, and here you have it. And these blue arrows are pointing to cells that have a very flattened nucleus, and you can see the reddish eosinophilic stained collagen fibers. Up here is the papillary layer of the dermis, and this is a particular layer, and these cells are in the particular layer, and they're fibroblasts, and so therefore they do produce collagen. But if you go back to the choices, we did not give you a choice of collagen. We gave you ground substance. Okay. The next question is number 11. You're supposed to zoom in on cells within a dashed line in the virtual slide specimen, and the correct answer would be lymphocytes. But many thought it was either a mast cell, plasma cell, or 14% of the class thought it was a mesenchymal cell, and almost everybody chose one correct answer except for the 29% that chose the correct answer, lymphocytes. So here's the specimen. Now what you have here, you have a medium-sized artery and medium-sized vein. You have adipose cells, and if you zoomed out, you would see that this is it's very likely the hypodermis, although you don't see the epidermis, but it's most likely that's what it is. But that is not key to answering the question. These are the cells in question with the dark, round, heterochromatic nuclei. And the size here, you see here's a red blood cell, it's 7 microns, so it's very close to that, maybe 5 microns in diameter. And these are the two structures, red blood cells and lymphocytes, that you were advised to use in internal measurements within a specimen. And you see this bar here is 23 microns. So if we move to red blood cell over here, you see it's very tiny compared to that measure. But they're lymphocytes, and that's the way you recognize lymphocytes. Very dark, rounded, heterochromatic nuclei. The next question is 12, and you were presented 3 groups of lightly stained cells enclosed by a dotted line, and you're asked which of the listed cell types is the correct identification. And the correct answer were mucous glandular epithelial cells. Some thought they were multilocular adipocytes in brown adipose tissue. Others thought they were unilocular. And some thought there were serous glandular epithelial cells. So let's take a look at those. Well, here they are. Now, if these were multilocular, you would see a lot of little tiny round lipid droplets in here. And if they were indeed fat cells, maybe I could find a fat cell. See, that fat cells look like this. Very clear, usually pretty large, with the nucleus right at the edge, very flat like here, and maybe here. Okay, so we go back to those, and you see they do not resemble fat cells, either unilocular or multilocular. They're certainly not serous cells. They're classic mucus cells where the shape of the cell is visible, but it's very lightly stained, full of mucus, and the nucleus is flattened at the edge. So this is an acinus, and it has a number of acinar mucus cells. These cells out here are serous cells, and this is actually a salivary gland that you'll be learning about soon in a future part of the course. Okay? Okay, this is 14, question 14. 2 cells, the surface of this is indicated by arrows of this specimen, and the answer was facet cells. But some thought they were pseudostratified cell, others thought they were a squamous cell, still others thought they were transitional epithelium. Well, yes, it actually was transitional epithelium, but that wasn't the question. We didn't ask the tissue, we asked the cell type. So, this is the specimen. And the cells indicated here at the surface, what are they? If you increase this, these are the dome-shaped cells at the surface, and their correct name is a facet cell. You should review the lecture on epithelium and on transitional epithelium, and you will see that information. You will also see that in the WebMic study guide if you go to the transitional epithelium and study it. Okay, this question presented two large cells enclosed within dotted line, and you were supposed to examine the nuclei indicated by yellow arrowheads and the nuclei in the two cells and select a true statement about these cells. Well, the correct answer is they were synthesizing proteins at the time of fixation. But a large number of the class thought the DNA in the nuclei is condensed. Okay, so let's take a look at the specimen. Here are the cells, and these are the nuclei, and in the nuclei you have a nucleolus, and then you have a somewhat basophilic cytoplasm here, and these are sensory ganglion cells in the presence of a prominent nucleolus with a rather distributed DNA in the rest of the nucleus, euchromatic. That suggests that these cells were synthesizing protein. So the only correct answer were the cells were synthesizing protein at the time of fixation. Okay, this presented a specimen of epithelial tissue, and it was actually simple columnar epithelium. We accepted two possible type in answers, simple columnar epithelium and simple columnar epithelial tissue. The specimen presented, well, only about 28% of the class got the correct answer. This is the specimen, and if you enlarge that in the box, you can see that this is the limit of the cell up here, and this is the basement membrane here, and you can see these large oval-shaped nuclei, and there's only one layer. So that's the only possible type of epithelium that can be is simple columnar epithelium. Okay, the next question was question 19, and the answer was simple cuboidal epithelium. You can see the several correct type-in answers we accepted, and 55% of the class got this correct. So you're asked to identify or classify the tissue that lines the spaces containing a black triangle. So here's the specimen here. These are the black triangles. And if you, let's say this, you focus on this one, enlarge it, you can see this is the lumen. Here's the limit of the cell. It's about as tall as it is wide, and the nuclei are very round. These are all indicators of simple cuboidal epithelium. We can go to, say, over here. This is another example. Same features. A cell that is about as tall as it is wide, and the nuclei are round in a single layer. Simple cuboidal epithelium. Okay, the last question in Module 1, Part B, is the black substance indicated by the arrows in this virtual slide specimen is foreign to the body. Now that's the first clue. Any substance that's foreign to the body, you need to be thinking, first of all, of neutrophils or macrophages. And macrophages would be the most likely candidate, and that was the correct answer. We accepted singular, plural, and also histiocyte is another term. But about 50% of the class did not get the correct answer. So this is the specimen. There's the black substance. And so that's the biggest clue, is this black substance here would have to be located within macrophages. Okay, that concludes the review of the questions that were most missed in Part B of the Module 1 quiz.